What do you think is the handicap to apply this in solid tumors?
I think, first of all, there's not a clear antigen as strong as CD19 in the solid tumors. I think there's an issue of trafficking of the T cells into the solid tumor microenvironment. So I think those two are probably the primary issues. T cell receptor therapy has been tested in solid tumors. That's a slightly different setting than CAR T cells. A chimeric antigen receptor or CAR T cell does not require MHC matching in order, it doesn't require the MHC molecules to match, whereas T cell receptor autologous cell therapy is basically delivering a construct that expresses a native T cell receptor on the surface of the cells, but it has to be MHC matched. And so in that setting, we're starting to see perhaps some early glimmers of responses, but I think it's a little more complex in CAR T cells, because when you have a native T cell receptor, it can recognize antigens expressed and presented by MHC molecules, meaning intracellular antigens can be expressed and recognized. With chimeric antigen receptors, CD19 is on the surface. It recognizes a surface protein. And so that's one of the disadvantages to CAR T cells, is they can really only be targeted against things that are expressed on the surface of the tumor.
Let me just add that CD19 is also expressed on B cells, and we've learned through effects of rituximab and others that most of us can live without B cells and do pretty well. So many of the antigens that are expressed on solid tumor cells will also be expressed in other healthy tissues, and how to activate a really potent immune response that can eliminate every last cancer cell without doing damage to healthy tissues is a much bigger problem for most antigens other than CD19.
So the first monoclonal antibody for the treatment of cancer was rituximab. It was developed in 1997. That built upon decades of work, from the original mouse antibodies that were developed, the B1 antibody, and eventually led to the chimeric antibodies, and now we have humanized antibodies. A lot going on in the field. What are your thoughts?
It's really interesting. You alluded to this, and when you put in perspective Ron Levy's lab, who had done a lot of work on, and Kohler Milstein, who had actually a Nobel Prize for the fusion of clones, and to try to deliver the first monoclonal antibody, right? A monoclonal antibody, for our audience, is an antibody that we can direct the specificity that's very critical, because it's a great tool. So it was developed in the mid-90s and approved in 1997 in relapsed refractory indolent lymphoma, follicular lymphoma, and was part of this trial. And then this was a trial that was initially also conducted at MD Anderson and Memorial, and it was very interesting to see patients who had very durable response. So then we started to, as we are now with the CAR trying to understand better, we have understood much more about the antibodies, although we don't exactly understand completely which mechanism is involved in the given situation. So to summarize for our audience, the mechanism of antibodies is to engage the immune system to try to fight against the lymphoma cells here, because it's an anti-CD20 rituximab expressed at the surface of all mature B cells. And then so there's three main mechanisms. The first mechanism is essentially antibody-dependent cellular cytotoxicity. So through binding of the Fc receptor, it attracts effector cells, NK cells, T cells, that can actually kill efficiently the lymphoma cells, CD20 positive cells. There's complement-dependent cytotoxicity that is to some degree obviously demonstrated, and we're not exactly sure in the clinic how important it is. Might be more important for ofatumumab, another antibody. And then there's also evidence of, in a preclinical setting, and probably from the clinical data, when we see the combination, the results of the combination with chemotherapy, that may be an anti-cellular effect, an anti-tumor effect by modulation of the BCL2 pathway and making the cells more sensitive to chemotherapy. So all of this, without overlapping chemotherapy, toxicity with chemotherapy, became an obvious tool to try to combine with chemotherapy. And that's what really changed the field in B-cell lymphoma across the board, indolent, aggressive lymphoma, etc. So in large cell lymphoma, the last real innovation in large cell lymphoma is R-CHOP, presented in 2000 by at the time the Gila, now Lisa group, and also the SWAG study at the same time, and the ECOG study that showed that R-CHOP, rituximab plus CHOP, clearly improved the outcome initially in more than 60 years old, and then, although now new 60 is 40, but over 60 years old, and then obviously demonstrated afterwards by a number of groups around the world that the clear benefit in the CR rate, PFS and overall survival that is maintained over time. So this has changed and set a new standard in large cell lymphoma, and we are still trying to make progress since the R-CHOP, almost 20 years ago. And so that's in follicular lymphoma. This has been also very critical in follicular lymphoma because we talked about the fact this is indolent disease. There was a lot of discussion, should you treat chemotherapy, because with chemo, you could sort of kill the good guys, right? And patients have shorter and shorter duration of response. And then with rituximab and chemo, you can induce much, much higher CR rate. And because by itself, follicular lymphoma is really highly responsive to rituximab, about 85 response rate, but the CR rate is 20–25%. It's not very high. As I mentioned before, a subset of patients that go into CR and get a small short consolidation can do really well, but typically that would not be sufficient. So the next step is that, how can we build up? So the next step after doing our chemo, and then was to use this as a maintenance. And the maintenance rituximab has been studied enormously across the board in all B-cell lymphomas. There's no evidence in large cell lymphoma that maintenance is beneficial if you have received our chemo, obviously. And in follicular lymphoma, there's no question, and probably in some situation in mental, it improves the PFS and duration of response, maybe the survival in mental in some patients who are responders to R-CHOP. I would actually question this. This is really a small number of patients from the original large trial, selected from the double randomization process from the European trial. But this is really something that has been an enormous tool and has changed the field in B-cell malignancies.